that I think that at the JCB, we're really trying to not just publish papers, not just sort of be the middleman between the lab scientists and the audience that's out there. We're really trying to be part of the community, of the cell biology community, and make a contribution to the community and to the field, and also determine a little bit and contribute to where the field is actually going. I think we've been doing this for quite a number of years now. And we're doing it in different ways, and one way is by being involved in policy issues, and as Emma alluded to, we have been involved quite heavily in particularly publication issues and data integrity policy issues. But another way of being involved in the community and contributing is actually to highlight important directions that the field is going to. And I think this meeting is essentially an example of that, because I think the topic of this meeting, cell biology of disease, is really a rapidly emerging and I think incredibly area of cell biology. I think it's becoming clear that if we really want to understand diseases, we need to understand them at a cellular level. It's basically not enough to just identify a mutation in a particular disease and know what the molecule that is affected, what the identity of that molecule is. It's actually important to understand what that molecule then does in the cellular context. And I think we're historically probably sort of for the first time in a position to actually do that really well, because there is so much fundamental cell biology out there that has essentially accumulated. We know enough about the basic cellular processes that we can essentially go into disease cells and ask, how are these disease cells different from normal cells? And also vice versa, as disease genes are being identified, we can now go very, very rapidly from the affected gene to a cellular pathway and a cellular process. And so this actually shortens the time between gene discovery and therapeutic applications, for example. And I'll give you one example, and of course it's from a disease that I'm working on, which is a premature aging disease called Hutchinson-Gilford progeria syndrome, where we have gone from identification of the gene to a clinical trial in 4 years. And that's really quite remarkable. And the reason why we could do that was basically because when the gene was identified, we could essentially tap into a very large basic knowledge of that affected protein. And I think that's really going to be a paradigm for the future. So I think we're going to see more and more of that